Okay, we're going for part 2 of chapter 5 here. So we're looking at, I think it's slide 15 in the PowerPoint, but anyway. Okay, primary versus secondary antibody response. So we're looking at how IgM and IgG, the actual antibody level in the serum that we look at. So let's describe this chart here. So when you first get exposed to the immunogen, see we're using that term immunogen this time. That means it's something, an antigen that we're able to produce an immune response to. So there's a little bit on the first exposure, the primary response. There's a lag phase. In other words, for like 7 to 10 days, no antibodies are being formed. So the innate system's doing its duty. So the first antibody that is made is IgM, followed quickly by IgG. And you can see it's almost equal amounts that are there. Then the IgM tails off and the IgG sort of tails off. Now, if you get re-exposed to it again, the lag phase is much shorter, just a day or two, as opposed to the lag phase at the beginning, very short on the secondary immune response. And you see how the IgG is like so much more robust response. The IgG is really increasing the secondary response, while the IgM is identical to the first response. So that's the whole thing with antibody titers. We can tell if a person has got their first exposure, because IgM and IgG are about the same, or their second exposure. IgM, is elevated, about the same, but IgG is definitely big time increase. So that's the whole purpose of antibody titers, to see if we have a primary or secondary immune response. And see, that's what the whole deal is with vaccinations, is that we're going to go through this primary immune response here and have the antibodies, so that when you get exposed to it again, then you're going to produce a lot of antibodies extremely quick. That's the whole purpose of it. I mean, you might still get a little bit sick, but you will have an antibody response that will, between the innate and adaptive, should take care of it pretty quickly. I mean, you still have some ill effects, but nothing compared if you didn't, as opposed to here in that primary immune response. So, antibody specificity and diversity is actually based on what we call clonal selection and immunoglobulin genetics. So you can see in the 50s, Jordan and Burnett had a theory about clonal selection on how we make antibodies, that lymphocytes are being pre-programmed genetically to produce one type of immunoglobulin, and that there was a specific antigen that looks for a particular cell so that it will respond to it and it will proliferate. The problem with that is you got to have a gazillion genes to handle that kind of nightmare that goes on. I mean, just think about it. You got to have an antibody for every single antigen that you might come in contact with. So, in 65, Dreyer and Burnett came out and said that there is constant and variable portions of immunoglobulin genes out there that are actually coded by separate genes. So in 87, Tawaga came out with a specific immunoglobulin gene segments that are selected and are actually joined as B cells start to mature. So we have what we call clonal selection. In other words, each B cell has a B cell receptor, that's the BCR, specific for a particular antigen. That was the initial clonal thing out there that we thought of. So when an antigen enters the body, it binds only to the B cell that has that particular B cell receptor specific to it. Okay, so that's what this picture is showing. So only those B cells that have been selected, that have attached to that specific antigen, will proliferate and produce antibody producing plasma cells, or it will make memory cells. Both, really. Okay? Now, immunoglobulin genes, of course, they're on the chromosomes. So the immunoglobulin genes are found on 3 clusters inside of different chromosomes, 14, 2, and 22. 14 is where the heavy chain is, 2 is where the kappa chain is, 22 is where the light chain is. And, I'm not particularly big on remembering that kind of stuff. Just know that these immunoglobulin genes are found on different chromosomes. So what will happen is we have rearrangement of the genes that is needed for us to produce a functional antibody. So we got 3 different chromosomes that are needed for us to produce a antibody. All right. So more than one gene controls the synthesis of a particular type of immunoglobulin. So the H chain, remember there is a variable region, so we have variable H, D, and J, which we'll talk about here in a little second. Then we have constant regions, which are controlled by the C genes. The light chains are controlled by the variable light, the J and C, so they don't have the D like we had up here in the heavy chains. So through just random selection during B cells as they go through their maturation sequence, these segments are joined together and tells the B cell, this is what antibody you're making, and this is what the specificity is going to be. So it's a lot of combination, a lot of combining that's going on through that. So here's showing you a heavy chain gene arrangement. So here's the V's, the D's, the J's, and the C's that we're talking about in the DNA. So you can see how the D and J join together. See how we have this gene rearrangement going on? And then the V's and the D's and the J's sort of combine here. So then we get all this transcription and splicing. The messenger RNA goes out, and then it's translated, and then it produces what needs to be produced on there. So it's important to know that we have all these different genes occurring inside of the B cell to tell it what antibody it's going to be making and the specificity of it. So the light genes, remember, they don't have the D part, but they go through a similar light chain gene rearrangement themselves. So you can see the C portion is extremely small in this particular because it's light chain. So it basically goes through the same kind of thing. So we go through a gene rearrangement for light chains and heavy chains, which we just saw. So what are some of the additional diversities that happen with antibodies? I mean, where do they come from? So there's some junctional diversity, when the V, J, and D segments, they don't always occur in a fixed position. They sort of move around a little bit more. So sometimes we make different heavy chains, and they will combine with different light chains. So we have this, what they call a hypermutation occurs. So this mutation will occur during an immune response sometimes. I mean, it's sort of saying, okay, let's make the best thing we can because we want it to attach to this foreign thing much better. So what we're doing is what's called affinity maturation. So in other words, we want the antibody that is being produced to have a real strong bind to that foreign antigen. So it's custom fitting it. Just like when you get a key cut, you want it cut exactly right. I mean, that's what's happening here. We're allowing the key to be cut so it will fit the antigen. So we're allowing the antibody to rearrange in the genes so that it will better fit that antigen. The better it fit, the better way it will attach, the stronger binding. That's called affinity. So like a key, the better it fits, the better it unlocks a door. So the same kind of thing is going on. So that's why sometimes we get switching of the classes of types of antibodies, depending on what we're trying to destroy, the immune response. So a lot of times in the laboratory, what we use is monoclonal antibodies. So monoclonal antibodies are used to help us do very specific testing. So they are actually based on how B cells can genetically pre-program themselves for a specific antibody. So what they do is they get a single clone, a single antibody-producing B cell, and they mix it with a malignant myeloma, like multiple myeloma, a malignant myeloma cell, and that's called a hybridoma. So this was actually developed in 1975, which, this is amazing. This revolutionized how immunology was done. So that produces a monoclonal antibody. So it's a clone from one B cell, hence monoclonal. Now, if it's more than one B cell type, then it's called polyclonal. So it's just another term that's used. So here's showing you how you do a hybridoma production. So here, looking at that, they get a mouse, they immunize it with a specific antigen, they take out the spleen cells. Remember, that's a secondary lymphoid organ. they will mix it with malignant myeloma cells in a polyethyl glycol, PEG. And what will happen is these cells will fuse together and they will make the desired antibody we want. And then they can like grow it and grow it and grow it and grow it. So we get tons of it. So, in summary, so the basic structure of immunoglobulin is a tetrapeptide. So it's got 2 heavy chains and 2 light chains that are actually joined by disulfide bonds. There's 5 different classes of antibodies out there. Immunoglobulins, IgG, IgM, IgA, IgD, IgE. And then there's light chains, which are kappa and lambda. But it's the heavy chains that makes the G, the A, the E, D, and E. The light chains are kappa and lambda. So each immunoglobulin has constant and variable. Remember that picture. Make sure you know what that picture is so you can actually, label it correctly. There are variable regions, which is like the top of the Y, and then there's constant regions. The variable region is where we bind to the antigen. The constant region, which is the heavier part at the bottom, is responsible for its functions. I mean, is it going to bind complement? Is it going to attract phagocytic cells? And what's it going to do? Okay? And then the 5 different types of heavy chains, what we just talked about for the isotypes. So there are some minor variations in certain types of heavy chains, hence they're called allotypes, and can differ among individuals, but of the same species. Okay? Because we've used allo before. The variable portion of the heavy and light chain is unique to a particular immunoglobulin molecule, and that's called an idiotype. Okay, so that's just a little variation in the heavy and light chain, but it's unique to that particular immunoglobulin. IgG is a predominant immunoglobulin in the serum, so it binds to complement, it does opsonization, this antibody-dependent cytotoxicity, that's what that stands for, neutralizes toxins and viruses, and it can cross the placenta to give passive antibody production to the baby, or passive antibodies to the baby. Passive means that the antibodies are pre-made, they're given to the baby doesn't have to make them. They don't last that long, they'll last 6 months or so, but it is antibody in there for the baby, give it some immune status. IgM is the largest of all the immunoglobulins because once it's out into the bloodstream, it's in a pentamer, so it's very great in binding to complement, and that's just called complementation. That's the binding of the complement, and to agglutinate with antigens to allow the antigens to stick to it. So remember from our chart, it is big in the primary immune response, so it's the first antibody to appear, and it's the first antibody an actual infant will develop. IgA is a major immunoglobulin in secretions, and it's as a dimer. Remember, it has that little J chain, and it has that little secretion component to it as it goes through the epithelial cells into the mucosal area. So it's in all the portal of entries, urogenital, respiratory, GI, to protect us from anything invading. So we got innate and we got IgA right there, right on the surface. It's also in breast milk, so it transfers passive again immunity to the newborn infant in the breast milk. IgD, like we said, we don't know too much about IgD. We know it's on B cells, and we think it plays some big role in cell maturation. IgE actually binds to mast cells and basophils, so the IgE will land on the allergen. The FC portion, that bottom portion of the Y, will allow, it has a receptor so that the mast cells or basophils can attach. Once they attach, it'll squirt out its heparin and histamine. Also, it's good for eosinophils, so it can destroy parasites, worms, helminths, that kind of stuff. So the primary antibody response is our first exposure, and it takes that lag phase of a few days, 5 to 7, sometimes up to 10 days, really, before we can actually detect it. Because it needs that time to go through all that processing so that the lymphocytes can be activated and go through the, the antigen-presenting cells to the T cells to the B cell. I mean, it's a lot going on. I mean, we basically covered it a little bit in the previous chapter, so there's a lot going on for us to get that primary antibody response. The secondary is from the memory cells, because remember, in the primary, we actually make memory cells. So when we get exposure to it, the memory cells, oh God, I've seen this before. I know how to do it. It's quick, bit, and out it comes. So it's rapid, and you can see from the graph, very high amount of antibodies. So that's where the titer comes from, is we don't really measure antibodies in milligrams per deciliter or grams per deciliter. We measure it in titers, and we'll talk about that when we get into labs. So it's actually like a ratio kind of thing. Like it's a 1 to 80, a 1 to 160, that kind of stuff. So that's how we measure antibody response. So that secondary immune response, that antibody titer, is a lot higher, and because in particular it's IgG, it will last a lot longer. So the clonal selection is when we talked about how lymphocytes can genetically program themselves to a specific antigen out there. So the antigen binds to the receptor of the correctly programmed B lymphocyte. It will then differentiate and produce antibody producing plasma cells. It's pretty amazing when you think about it. So, but it's that clonal rearrangement that we were talking about. You know, because remember, there was a whole bunch of different genes, and we got to go through this random selection. So those segments will join to make an antibody of a single specificity. So they all sort of talk together, I guess you could say. So they know how to produce something that's very specific for a particular antigen. Then we have monoclonal antibodies. Monoclonal antibodies, remember, are made of hybridoma cells. So that is a one antibody-producing B cell fused together with a malignant myeloma cell, a plasma cell that's malignant, so that it will just produce like crazy. And they live forever, these malignant cells. I mean, it's hard to kill these things. So we get, from a hybridoma, we get monoclonal antibodies. In other words, things that are super, super, super specific, and work extremely well for us in the laboratory. So, like it says here, monoclonal antibodies are used for laboratory reagents. I mean, we use it like crazy. Just watch it on TV. Oh my God. I mean, there's gazillion therapies out there. Look at the fancy words, and it has a little MAB on it. Guess where that MAB comes from? Monoclonal antibody. There's actually a video in your content on D2L that will talk about this. So look at that video. It's pretty slick on when they're talking about what monoclonal antibodies can do. Okay, so that is Chapter 5. I did it in 2 parts just because I need to take a break myself, but make sure you read the chapter. Look at that summary. It does an excellent job in the summary and the study guide. You know, gives us nice bullet points. There's some case studies in there, and there's review questions. Very good review questions. Okay, and the answers are in the back of the book. So we're in Unit 2, Chapter 5. So if you have any questions, make sure you reach out, email, text, see me in the lab. Hope you enjoyed it. Thanks.